Sartorius is a leading international partner of the biopharmaceutical industry. From discovery to process development to clinical and commercial manufacturing, our portfolio of proven solutions helps our customers achieve faster time to market while saving costs. Together, we're setting the standard in cell and gene therapy. Hey everyone, welcome to episode 91 of Cell and Gene Podcast. I'm Erin Harris. Thanks for tuning in. You're about to listen to my conversation with BlueRock Therapeutics Dr. Amit Rakhit. Amit's a newer member of the BlueRock team, and his upbeat demeanor is nothing compared to his passion for the treatment of Parkinson's disease, a progressive neurological disorder that affects movement. But as you'll hear in just a sec, among other aspects of Parkinson's treatment that he and I get into, Amit explains that how by injecting and implanting live pluripotent stem cells to allow for dopamine-producing brain cells that were lost has led to improvements beyond motor function, whether that's around sleep or mood, those that are not addressed by just traditional dopamine replacement therapy. I'll let Amit share the entire story, but suffice it to say that the potential of BlueRock's therapy is that they integrate back into and create the neural network that was lost with the pathophysiology of the condition. Amit and I also talk through how cell therapies might offer a different approach to long-term symptom management for PD. And we also even get into manufacturing. It's a really great episode, so settle in and enjoy my discussion with BlueRock Therapeutics Dr. Amit Rakhit. Hi, Amit. Thanks for being here on Cell and Gene, the podcast. Welcome.
Great. Well, thanks for having me.
Good. Tell us a little bit about BlueRock Therapeutics. I know you are new to your role, so tell us what you have been doing since you joined the team.
Sure. Well, I'm super excited to have joined. I joined this summer in July of this year at BlueRock Therapeutics as the Chief Medical Officer. Chief Development and Medical Officer is my official title. Really joined the company because BlueRock is leading the field in terms of cell therapies as potential new treatment options for patients who need them. I've been in the industry for more than 20 plus years now, having spent time in large organizations. I started as a physician, of course, and then I moved into the industry about 23 years ago and started at Bristol Myers Squibb. So I have a long history of small molecule development. My original drug was Plavix that I worked on as part of the original development team, and then moved into smaller companies, including Biogen, where I helped on the Spinraza program, and then smaller companies even after that with Ovid Therapeutics, Flare. And then what attracted me to BlueRock was that really cutting edge science, the search and innovation, looking for what could be possible. When I went to med school, cell therapies and gene therapies, those were possibilities, but not reality yet. And here I joined because that possibility is becoming a reality today. And that's really exciting to see that happening. Plus, the team is super engaged, super smart. I love working with lots of smart people who know a lot more than I do. And if I can help kind of patients who need those new therapies, and we can help kind of push the needle and envelope on this field, that's why I joined. I think we're doing so with our new therapies. I would say BlueRock is really focused on a couple areas right now. Our lead program is in Parkinson's disease, so in the neurology space, with some follow-up programs as well. We're very excited about that program moving into its next phase of clinical trials. And then we're also in the ophthalmology space with inherited retinal diseases, and that we just received clearance for our IND by the FDA just recently. And that's also starting a clinical phase 1 program this year. So 2 clinical programs, plus a huge amount of research efforts that are really looking at other areas that we can actually think that cell therapy has a place for.
Good. Well, thank you for all of that background information. And I'm sure that your colleagues at BlueRock are just as happy to have you there as you are to be there. So really exciting all around, I'm sure. You did mention Parkinson's, and that's what I want to talk about first. So we know that there are some limitations to current Parkinson's disease medications. And, how might, say, cell therapies offer a different approach to long-term system management, even especially as the disease progresses?
Sure. Yeah. The Parkinson's is, neurologically progressive condition. Um, Yeah, as you have deterioration of the specific neurons in the brain that produce dopamine, right? That really affects motor function and probably even beyond motor function, non-motor function as well. But the mainstays of therapy have been dopamine replacement therapies, where you can take an oral pill and start to alleviate some of those motor symptoms that you start experiencing as your condition progresses. There hasn't been any really novel innovations in the Parkinson's disease treatment landscape for the past 60 years. Most of these have been either dopamine replacement therapies, better alternatives that are still dopamine focused, long-term infusion. And I think the cell therapy space, gene therapy space, the whole regenerative medicine space offers a really interesting alternative. That's a therapeutic option that people are looking for. So when you speak to physicians and clinicians who are treating people with Parkinson's, there's limitations. You're looking at dopamine therapy to start with, but that effect wanes over time. Typically about 5 years or so, you're seeing people who are either taking more frequent pills because their symptoms keep recurring, so they have to increase the dose, or they have to increase the frequency of their dopamine therapy until it really is not working for them anymore. So then what do you do next, right? Then you do more advanced therapies. There are continuous infusion dopamine pumps. There are deep brain stimulation as a surgical procedure. And there's not much else right now. So looking at this space, looking at that high unmet need, we see that cell therapy is a potential option. So our lead program is bemdaneprocel, which is DA01. It's a human stem cell derived progenitor for dopamine producing neurons that is surgically placed into the putamen area of the brain. This is the area where you see the degeneration of those dopamine producing cells that lead to the symptoms and the overall disease pathology of Parkinson's. So it's a surgical placement under anesthesia, where we actually have grown these progenitor cells. And we take these stem cells and grow them in media so that they differentiate down a neuronal path. And then once they get to a sufficient level of maturity, we're able to place those cells into that area where the degeneration of these native neurons have happened. And at least in our clinical work, we have a phase 1 study that read out for just recently, and we presented data at some of the major conferences. We've had 18-month data as well as 24-month data. You see improvement. You see improvement in what we call the on state. So looking at assessments for Parkinson's. For those people who are not familiar, there are kind of standardized assessments for Parkinson's disease that we can look at. And there's some good time, which is called the on time, when people are not symptomatic. And we see an improvement there, approximately 1.8 hours at 18 months, and then a decrease in their off time, the off time being those times when they're symptomatic and they're not doing as well, and they can't have their daily functions of life. And we see a corresponding decrease about 1.9 hours. And you may think that, hey, 1.8 hours, 1.9 hours, is that really clinically meaningful? It actually is, right? Think about somebody who is now able to go to a restaurant, have dinner, come back, and know that they'll be okay for those 2 hours that they're out, right? Or being able to sit through a movie and, with a loved one or a significant other and come back. So I think these changes are meaningful from not only a person who's experiencing Parkinson's disease, but their surrounding kind of support circle and their family and their loved ones. But also when you speak to physicians, that's clinically meaningful. When we see these changes that are going to make an impact for somebody's daily life, and that's especially in a neurodegenerative condition, which you expect to progress, having improvements when typically you wouldn't see improvements, you would see further decline. That's something clinically meaningful. Now, I'll just caveat that by saying we had a small study. It was 12 patients in this phase 1 program, but we're really encouraged by that data. We're really encouraged by the feedback we've received from clinicians. We're encouraged by the feedback we've received from the patient advocacy organizations saying, look, we need this new therapy. You guys are, on the cutting edge of that. How exciting. And even further to that, we have regulatory interactions. We met with the FDA, and they actually granted this program a designation called RMAT, or Regenerative Medicine's Advanced Therapy. And very few compounds get that designation. It's really meant for programs that the agency thinks can have a meaningful difference and bring a new therapeutic option for patients. So very excited about the program moving forward.
Good, good. That's wonderful. It's so wonderful to hear about the time that the patients, whether it's slightly over an hour, that they're able to
almost 2 hours, not quite.
Almost 2 hours.
Almost 2 hours.
Feel. I certainly would never want to speak for a patient, but I suspect that you've talked to them and they're happy to have that time to feel somewhat normal, and be able to go and function outside of their home or whatever they choose to do.
Absolutely. I mean, I actually have a personal connection to that I had a time when I was at one of my other companies. I was able to live in Paris for about 5 years or so, running the division out there. And one of my very good friends was diagnosed with Parkinson's disease. And I saw him over the course of 5 years just progressively deteriorate. He was an amazing fashion executive at the time, had this amazing, interesting life that you just kind of read about sometimes or see in movies. But over time, his symptoms worsened. He, would come over to the apartment, and I remember he had such difficulty in just reaching for that little bit of food on the table, that piece of cheese, and bringing it up, and the shaking in his hands, when you knew that, mentally he was fine, but those motor symptoms really impacted the way he interacted with the world. Sure. And that was something like I still remember because I saw him, when I first got to Paris, and then 5 years later, that progressive decline. He didn't want that, right? Nobody chooses to be ill. Nobody chooses to have these conditions. But when you do, you kind of rise up and you try to do the best you can, and you take it day by day. And he did that. He did that amazingly well. But I remember that, and that's one of my reasons. When really saw this program, I say, wow, this is a chance to impact, an individual, maybe a lot of individuals, hopefully, who have to deal with this day by day.
All right, thank you. So bemdaneprocel, talk to us a little bit about how its MOA differs from the traditional PD treatments.
Sure.
And, we know the advantages, but what's different about it from what we have now?
Sure. So right now, when you actually take one of the dopamine therapies, it's a small molecule. You're actually replacing dopamine by just taking a pill, right? So this is dopamine replacement therapy. What we do with bemdaneprocel, what happens in Parkinson's is that you're having degeneration of those neurons that produce dopamine in that area of the brain, the striatum, actually are degenerating and dying. And so you no longer can produce native dopamine from the cells that are supposed to produce dopamine that help as one of their functions coordinate motor function. So we're actually injecting and implanting live cells. These are live pluripotent stem cells that are able to differentiate into whichever cell type we'd like them to go. And through the talent and brilliance of our research team, who are able to really grow cell cultures and provide a nutrient media with growth factors and proteins and other aspects to differentiate down a neuronal pathway, that's what we actually implant into the patients with Parkinson's disease. And so these progenitor cells then further differentiate once they're implanted to become those dopamine producing brain cells that were lost, that the individual lost. So it's not only providing dopamine as an oral therapy, you're actually creating those and replacing those brain cells that can produce dopamine. But what they also do is they form this neural network. So they start to create and recreate the neural network that has been lost. So beyond just the motor function that these neurons actually are very important in, there are likely some non-motor functions as well. Whether it's around sleep, whether it's around mood, whether it's around other aspects, those are not addressed by just your traditional dopamine replacement therapy. These are more inherently part of the neural networks that are created. So the potential for our therapy is that we integrate back into and create the neural network that was lost with the pathophysiology of the condition.
Gotcha. Okay. We know that Parkinson's disease is the second most common neurodegenerative disorder next after Alzheimer's. So talk to us a little bit about, from your perspective, the impact of urgency for developing new treatments like BlueRock's. Where are we with going forward with it?
I've been in development for 23 plus years. I think when you hear from people who are affected by illnesses, when you hear people who are infected by condition like Parkinson's disease, the impact it's had on their life. This is a condition that hits people in, kind of the mid kind of prime of their lives. People are in their 50s, 60s, when they're getting diagnosed with this condition, when you're just about to hey, I've worked all my life, I'm about to enjoy, I'm just gonna and do things, and you've got this life experience, and then you get hit with something like Parkinson's and diagnosis, and it changes your life. It just upends it. So, and like as I mentioned, there hasn't been really revolutionary or really many changes in that therapeutic landscape for the last 60 years. It's all been around dopamine because we just haven't been able to get there. When I was in med school, like I said, it was kind of science fiction to be able to replace brain cells, replace living cells into when they're lost back into an individual. And so we're getting there. We're getting to that treatment kind of evolution that's wow, we can actually do something that's pretty amazing that was thought not possible before. It has its challenges, right? So these are cells that are not your own cells. So you have to be on immunosuppression. And right now our immunosuppression regimen is about a year. That can have some challenges too. But we see that there's this, even with our small number of patients, you see this, change in function. That's an improvement. That's maintained 18 months, 24 months. We're going to follow people longer as well. So you're coming out to almost 2 years that you're actually maintaining that effect we see when traditionally, if you had not intervened, had no therapies, you would see a progressive decline. And I think that's really amazing to see. So when you see encouraging data and trends, it just kind of spurs you on to think that, yeah, this is something that we need to pursue. Patients and people who are affected by illnesses are waiting, and every day counts. So that urgency is there. I can feel it definitely at BlueRock. We're very excited about the programs, and it's across the industry in biotech, right? When you're in that innovative cycle, we want to be able to go beyond just the research, beyond the development that we do. It's really all about people and patients and, really curing illnesses. I mean, that's why I became a physician, is because I'm here to help people and I want to do that. And so, yeah, if I can bring a little bit of spark, a little bit of drive, and keep that momentum going, whether it's internally, whether it's having a conversation with you to get the awareness up, whether it's talking to regulators, all of us have a part to play in this, and it's amazing that we can do so. So yeah, the urgency to treat is always going to be there, and I'm always going to help drive and hopefully drive that forward.
Good. Love that. Talk to us a little bit about the Expedite trial. So what have been to date the most significant findings from your 24 months worth of data?
Yep. Yeah, it is. So, so I kind of mentioned that earlier. So this is this trial I was referring to. It was called Expedite. This was an open label, so it's not placebo-controlled. It's a phase 1 study, and typically in phase 1 studies, you're really understanding safety in this population, but we also look at measures of efficacy or see if anything's working there. We did 2 cohorts. There was a lower dose group, about 5 patients, and then a higher dose group at 7 patients. And it's in the higher dose arm, which we're planning to move forward into our next stage clinical study, is that we see these differences from baseline, these improvements in what we call on time, the 1.8 hours that I mentioned, as well as the corresponding decrease in off time, which is the not so good kind of hours of 1.9. So that's the study that was conducted. Like I mentioned, it's open label, so it's not controlled. However, why do you need typically a controlled study, which is what we'll do in our next study, is that you can account for placebo effect, right? Sometimes people, just by being in a clinical trial, will feel better or do better. And so you have to account for that. But as I mentioned, there's a couple of things here that are interesting. First of all, we do see a difference between the low dose and the high dose, with the high dose having trends to more improvement. So that's something that speaks against, let's say, quote unquote, placebo effect. The second is typically people who have a condition that's degenerative, right? That's going to progress over time, that you're going to actually deteriorate over time. That placebo effect, that feeling like, okay, I'm doing better, is going to wane. And typically that waning happens within that first year, right? So it's variable. Some people will say 6 months, 9 months. Let's say within the first year, that placebo effect should really have gone away, and that you should start to see the natural history take over again in terms of the decline. So now that we're seeing actual improvements at 18 months, which is maintained at 24 months, that's very encouraging as well. So I think that's part of the reason that we've had, good interactions with our physician community, patient community. They see the natural history with people, so they can understand that, hey, this may be something interesting here. We've had interactions with the regulatory agencies, specifically FDA. They're very encouraging as well. They see a lot of programs. So I think that's, also for me, very encouraging. So there are signs and trends that are really pushing us into the direction like, hey, this might be something really meaningful, and we need to pursue this, and really, and really give patients an opportunity to, that they need.
Good. Okay. Can you discuss any plans for the trial? What's next?
So, like I mentioned, we've had interactions with the FDA. Our plans are to initiate the next stage clinical study, which will be a larger study. The first study was in the US, so it was only, like I mentioned, 12 patients total. This will be a much larger study. It's still kind of being looked at right now, but it's probably closer to that 80 to 100 number of patients or participants who choose to be in the study. So there'll be 2 arms for this study. There'll be the active arm where you would actually receive bemdaneprocel. And then there's the sham placebo arm, because we do need to understand, if you don't get the therapy, what happens. It's a 2-to-1 randomization, so there'll be more people getting the active arm, so about twice as many as the placebo arm or the sham arm. It's a surgical procedure which is done in the OR under anesthesia. So that is something to consider when you're considering participation, is that it's a surgical procedure. But during that surgical procedure, we implant 2 burr holes, or we put 2 burr holes into the skull, and then actually implant into the region, as I mentioned, where those cells are lost. So it's in this putamen area on both sides, on the right side and the left side. We put in about 3 million cells per side. and then come out, and then we follow the participants from their baseline all the way through multiple assessments to about 18 months, right? So 18 months, and then they roll into a open label extension part of that study. So at that time, people who are on the placebo will have the opportunity to then actually have a procedure again to put the actual cells in, so they're able to have the active cells once we assess whether it's working and whether we believe that the benefit risk is there to continue development. So there's a double-blinded phase followed by an open label phase, and the primary endpoint is around 18 months, at 18 months.
Nice. Okay, well, we'll be following up with you too after the next steps of as the
Sure.
triathlon goes.
Yeah.
So
We're very excited to start. I mean, we're in process of actually submitting our documents to the FDA so that we can actually start that program, hopefully, with early next year. So this is what, October? No, almost November. So really, we're on track to start in 2025.
excellent. Before I let you go, we cover manufacturing extensively on Cell and Gene. And so I'm always interested to hear from my guest's perspective, if manufacturing is part of the equation, their thoughts on it. And so what were, say, the key challenges in manufacturing is, in delivering the allogeneic cell therapy? And if you ran into any challenges, and I get it, people always do, how did you address them?
Yeah, I think I've been more in small molecule and oligonucleotides, so definitely the cell therapy space has definitely opened my eyes and perspective on the challenges of manufacturing. And like I said, the super smart people who know how to differentiate these pluripotent stem cells into whatever cell you're looking for. So it's such a huge respect for that knowledge on biology and but it is biology, right? So there are sometimes unexpected events, and you have to account for living cells. I think one of the areas of interest that we're always looking at is making sure that the cells are differentiating down the proper pathway, and that there are no potential concerns for teratomas, mutagenicity, and there are markers for that, for example, like Ki67 and other proliferation markers that we look for and try to minimize. So what is that right balance? So I think understanding that sometimes you need those proliferation markers to be high for a certain point of time because you want those cells to differentiate and grow. But at some point, they need to come down to acceptable levels. And I think that's something we're always looking at is like, what are cell proliferation markers? How do you get consistency in actually developing? So when you're doing a study for 12 people is one thing, or 100 people is one thing, but then thinking down the road in the future, if we're successful, and we hope we will be, how do you then manufacture for thousands of people, and how you ensure the consistency that's needed when you're growing living cells? And I think that's something. What's the right immunosuppression therapy that you think about, when those cells are, which markers are there? Can we optimize so that you have less immunogenicity? I think that's, again, still a little bit something working on in terms of the future. I think that's another area that people are interested in for cell therapy. But I think the challenges around proliferation are challenges around consistency. When do you introduce automation? How do you scale up? Those are all, I think, part of the dialogue. I know with my CMC colleagues all the time, but as it's part of development too. And I think we're all integrated in a way that we're thinking about these challenges each and every day. And every day there's like something new that I either learn or that we're learning, or a challenge that we've overcome, or a new challenge that comes up, right? So it's kind of evolving, as you know, then as the cell and gene therapy is such a vibrant, dynamic space that is evolving every day to something that we're learning more about this space. And I think it's going to be almost like how oligonucleotides were when they first came out, right? There was this breakthrough, and all of a sudden, there's so many people in this space. it's kind of like that now for cell and gene therapy. We're learning as we go, but we're making a lot of progress. And I think in the cell space, I think some of those are the manufacturing challenges I mentioned, but I think you could say there's challenges in just about every area.
That's a fair statement for sure. But you're right. We've, even just as early as 5 years ago, I think it was a different space than it is now. And with each passing year, new certainly challenges come up, but the evolution of it outpaces any of the challenges times.
Yeah. What is that? Something like a step forward, 2 steps back, sometimes a little zigzag, and you keep on moving forward. So something like that.
That's right. That's right. All right, that's a wrap on this episode of Cell and Gene, the podcast. Thanks to BlueRock Therapeutics Dr. Amit Rakhit for his time. Amit, it was great to have you as a guest on the podcast. Thank you so much for your time and all the insight you shared today. This was wonderful.
Awesome. Well, thank you so much for making the time for me. And yeah, definitely to your audience, any questions, please reach out to us at BlueRock. Happy to answer, and would love to keep in touch, Erin, as we progress. Would love to kind of share more news as we generate it.
Oh, for sure. Well, we're following you, always have been. All right, listeners, check us out at cellandgene.com, and we'll see you next time. Talk soon.